Modern neuroscience has become incredibly proficient at studying how brain circuits function. Using tools like optogenetics, researchers can place behaving animals in virtual environments and use specific colors of light to directly activate or deactivate individual neurons. Yet, a strange paradox exists in the field. When scientists study the function of a neural circuit, they look at the cellular level. But when they study how that exact same circuit develops and assembles, they drop entirely down to the molecular level. This reveals a severe mismatch in how we approach biology. Information processing in the nervous system is handled by living cells, but when we look for the instructions on how those cells wire together, we mistakenly assume we can read them directly from the genes. Simply put, genes encode molecules. They do not encode neural circuits. Confusing these two distinct levels of biological organization breaks our ability to understand how complex structures actually form. This confusion stems from the blueprint assumption. We frequently treat the genome as if it contains a pre-drawn, literal map for wiring the hundreds of thousands or billions of neurons in a mature brain. By attempting to reduce the architecture of the brain to a simple genetic schematic, research falls into massive category errors. We assign high-level architectural properties to microscopic chemical chains. You see this error most frequently in the public and scientific interpretation of behavioral genetics. When genome-wide association studies process massive amounts of DNA to find a trait, the media inevitably reports the discovery of a gene for that specific behavior. If we want to understand how a brain is built, we have to stop reading the genetic code as a final blueprint. Instead, we need to look closely at the autonomous agents, the growing, moving cells that are actually doing the construction. Take the visual system of a fly or the human cortex. In their mature forms, these regions exhibit highly organized, morphologically distinct layers where specific types of neurons make precise connections. Looking at the finished structure, the intuition is simple. A growing axon must run a genetic program that instructs it to find its specific pre-existing layer and plug in. The biological reality is entirely different. At the time most of these axons are pathfinding, those target layers do not exist. Instead of finding a target, the axons build it through algorithmic growth. A pioneer axon stops, establishing a boundary. Subsequent axons arrive, positioning themselves relative to whatever is already there. Successive waves intercalate, generating layers chronologically through physical interactions. The structures of the brain are generated in real time by the active growth of cells. Because the targets themselves develop in a concerted ballet of feedback, the genome cannot possibly hold a static map of the final product. Because we ignore this intermediate level of growth, we routinely mislabel individual genes based on higher-level outcomes. Consider the well-known protein DSCAM. In biological literature, the gene that codes for DSCAM is widely classified as a self-avoidance gene, but functionally, that phrase is an oxymoron.
At the strict molecular level, the DSCAM protein performs exactly one function: homophilic binding. If you isolate these molecules in a dish, they act as an adhesive. They stick specifically to themselves. Self-avoidance is a cellular property. It only emerges when this sticky protein is deployed on the surface of branching dendrites. When two branches from the same neuron bump into each other, the DSCAM proteins bind, triggering a cellular mechanism that forces the branches to recel. The gene does not contain the rule of self-avoidance. It merely provides the chemical hardware. The rules are executed by the cell, in context. Scale this logic up to the entire organism, and you begin to see why genetic data consistently fails to predict complex human behavior in modern association studies. In 2019, an enormous study analyzed the genomes of nearly 500,000 people to find the genetic basis for sexuality. They found no single gene. In fact, using the entire genetic code, researchers could predict less than 1% of individuals' same-sex behavior. Similarly, a major study of monarch butterflies claimed to isolate a gene for migration. Upon closer inspection, the sequence in question turned out to be a gene coding for a collagen required for muscle function. The non-migratory butterflies simply needed less muscle. This chart shows why genetic correlation isn't cause.
In the bottom graph, deterministic genetic instructions degrade through unpredictable developmental noise. Look at the top graph. The effect of a single mutation approaches zero, drowned out by thousands of other interacting genetic factors. Genes for specific behaviors are statistical illusions. A final trait is almost never dictated by a single variation in the genome, but by the massive, unpredictable interdependence of thousands of variables developing over time. This high level of interdependence explains a persistent mystery in genetics: why a seemingly severe mutation can have absolutely zero measurable effect on the vast majority of an organism's cells. Imagine a delivery vehicle on a city grid. Block the shortest route? It compensates by taking a fast detour. Limit its speed instead? It takes the direct route. But combine these defects, a slow vehicle forced onto a detour, and the entire system collapses. To a scientist mining data, these two genes appear redundant because a mutation in either one causes no obvious harm. The reality is that one mutation merely created the highly specific, sensitized environmental context that allowed the other to fail. We cannot predict the endpoint of an organism's behavior by simply reading the bedrock genomic code. There is no straight line from DNA to thought. Biological information is generated through time, space, and continuous environmental feedback. There is no mathematical shortcut to the final connectome. To know the outcome, the algorithm of physical growth must actually be run. Continuing to search for a gene for intelligence or a gene for alcoholism ignores the physical reality of how biological systems assemble themselves. If we truly want to decode the brain, we have to parameterize our research at the correct level. We must observe the cells as the active players and uncover the rules they follow in real time.